and I welcome you to our fourth lecture on the developmental evolution of female reproduction. And this time we will talk about the evolution of menstruation. In a recent review published in the American Journal of Obstetrics and Gynecology a few years ago, it was argued that even though menstruation is a normal part of the physiology of every woman during the fertile years, the medical implications are actually increasing quite dramatically. In the 1919, it was estimated that a woman during her lifetime had about 40 menstruations, while due to the availability of birth control measures and less lactation, there is now an estimate that an average woman experiences 400 cycles of menstruation in her lifetime. And that leads, of course, to a proportional increase with menstruation-associated disorders like abnormal uterine bleeding and so on, with huge consequences and burdens on society. So, given the importance of menstruation for the health and well-being of women, it is quite surprising that we actually understand very little about menstruation. Here is a review by one of the preeminent researchers in the area of the menstrual physiology, Hilary Critchley. In 2015, she wrote in her introductory paragraph, "The phenomenon of human menstruation has been shrouded in mystery throughout history. Many questions regarding menstrual physiology remain unanswered, not least, why does it happen?" Of course, why does it happen is a question that is fundamentally an evolutionary question, and I would like to pick up this challenge here. However, I'm not here today to give you a definite answer, because my estimation is that there is no answer. There's not a simple answer, not even a complicated one. What I want to achieve in today's lecture is to give you the outlines of the problem, such that it can clear the ground for further investigation into the question of why menstruation exists at all, what biological role it may play, and how we can understand it in general. Now, let's get the primary things out of the way first. I think everyone knows what menstruation is. I do give here a technical definition. It is menstruation is the shedding of the functionalis, that means the differentiated part, upper part of the endometrium, with associated vaginal bleeding after the luteal phase, meaning after ovulation and after the corpus luteum has ceased producing progesterone. Now, it is a broad consensus that the proximate cause of menstruation is luteolysis, so the degeneration of the corpus luteum, and with that a fall in the level of progesterone, which is followed by the invasion of pro-inflammatory leukocytes like neutrophils and eosinophils, leading to the tissue breakdown and hemorrhage and so on. And it is, however, important to remember that there are other forms of physiological vaginal bleeding that are not menstruation. For instance, one is often asked whether dogs have menstruations. They do have vaginal bleedings on a regular basis, but the vaginal bleeding in dogs is associated with the proestrus, or the time in the cycle before ovulation, and is due to the hyper development of blood vessels in the endometrium that sometimes can break through the surface of the endometrium and leads to extravasation and bleeding. So let's start with the comparative biology of menstruation. Before we go there, we remind each other of the major groups of mammals, because it is a mammalian phenomenon, of course. So there are three major groups of mammals: the monotremes, marsupials, and the so-called placentals, or better called eutherian mammals, because marsupials also have placentas, and therefore people are right in not calling eutherian mammals placentals. Monotremes are, as I often say in my lectures, are essentially hairy lactating reptiles. So in many ways, they are very similar to anatomically and physiologically with reptiles. They still lay eggs, but after hatching, they do provide lactation, but they don't have nipples and so on. Live birth most likely evolved in the stem lineage that leads to the common ancestor of marsupials and placentals, where the difference between marsupials and placentals is that marsupials have a much shorter gestation and a much more limited fetal maternal interaction time, while placentals or eutherian mammals are characterized by a much more extended gestation and the ability to give birth to quite advanced neonates. Now, if we look among mammals, menstruation is only found in eutherians, so excludes immediately the marsupials and the monotremes, of course. And there, it's actually only found in maybe roughly 150 species out of about 4,000 eutherian mammals. So that comes down to about 4% of the eutherian mammals are actually menstruating. The largest number of menstruating species are primates, humans and apes, of course, Old World monkeys and some New World monkeys, but not the other primates like tarsiers and so on. There's one species of Afrotherians, so animals that are related to elephants and dugongs and so on, and that is the elephant shrew. We have a rendering of an elephant shrew. Interestingly, there are 4 to 5 bats that have been reported to be menstruating. And there's one most recently described, one rodent species that is also menstruating, the Cara spiny mouse, Acomus caharinus. And I think the discovery of a rodent with menstruation was a major breakthrough for the whole field, because so far none of the menstruating species are attractive lab animals. Primates, of course, are highly problematic as lab animals, in particular if you want to have large numbers of them. Afrotherians, or elephant shrew, is also not, can be kept in zoos, but is not fast reproducing and a relatively large animal for a lab species. And of course, bats are not convenient lab species as well. So I think we are at a time in history where more comparative research on menstruation will be proceeding, and I want to give you an overview of the context in which this research will take place. So if we take this information that I gave you about which species are menstruating and put them on a simplified phylogenetic tree of eutherian mammals, you see here at black all the lineages of animals with menstruation. There are a number in the bats and one in the Afrotherians here and the rodent. And if you look at that long enough, you come to the conclusion that there are at least 4 independent originations of menstruation. So it's not just evolved once, but actually 4 times independently, which also leads to complications, because it might be actually evolving for different reasons. But for now, let's assume it's all the same phenomenon until we learn more about it. So then I would like to give you an overview of what the biological characteristics of menstruating species are. And I like to do that in the following way, by using a sort of an Venn diagram. So each of these boxes here represent a set of species. So the largest box here is the set of eutherian mammals, and the smallest here species with menstruation. This is this light red one. And so within the eutherian species, there are species with invasive hemochorial placenta, and it turns out that all species with menstruation are a subset of the invasive hemochorial placenta bearing species. And just as a reminder, there are these three different forms of placentation, depending on how many tissue layers are between the fetal blood and the maternal blood. The least invasive one is called epitheliochorial because the trophoectoderm epithelium is attached to the uterine epithelium, and there are at least 1, 2, 3, 4 cell layers separating the fetal and the maternal blood. The next more invasive form is where the uterine epithelium gets eroded so that one cell layer is removed between the fetal and the maternal circulation, and the most invasive form is hemochorial placentation, where the surface of the placenta is directly in contact with the blood of the mother. Now, and what is interesting about this classification is that if put on a phylogenetic tree, it is clear that hemochorial placentation is ancestral for eutherian mammals. The ones that are not hemochorial or less invasive, like all the hoofed animals, pigs, llamas, cows, and so on, they have secondarily evolved non-invasive placentation, epitheliochorial placentation. Now, interestingly, none of the epitheliochorial species are menstruating. That is significant because the hoofed animals are actually a relatively large group of species. In particular, the cloven-hoofed species, so that like cows and their relatives. Only think of the gazelles. There are many species of gazelles living, but none of them are menstruating as far as we know. So that suggests that species with menstruation are a proper subset of all the species that have hemochorial placentation. Now, within the eutherian mammals, there's also a difference in the mechanism of ovulation. There are some that have spontaneous ovulation. I will explain what that means in a second. And this is a subset of all the eutherian ones. And it again turns out that species with menstruation are fully contained within the species with spontaneous ovulation. So ovulation is, of course, the release of an ovum from the follicle in the ovary, and the stimulus that leads to ovulation can be different in different species. So there's environmental ovulation caused by temperature changes, light cycle, and so on, indicating the time of the year. You find this very often in fish, amphibians, reptiles, and so on, and also in some mammals, of course. Then there's this phenomenon of male-induced ovulation that can be either due to pheromones, as it is, for instance, the case in opossum, Monodelphis domestica, where the pheromones from the male induce ovulation in the females, or induced by copulation, either by mechanical stimulation or through some molecules that are transmitted with the seminal fluid, can lead to ovulation. That is, for instance, the case in rabbits, cats, and other animals. For instance, llamas have a chemical induction of ovulation. And then there is spontaneous ovulation. That means it's determined largely, but can be modified by environmental influences, of course, by internal endocrine mechanisms. And this is what we know, for instance, from humans and mice and so on. Here, just a cartoon to illustrate these different forms of ovulation. So the most primitive one, most widely found in all kinds of animals, is environmental signal calling causing ovulation. Copulation then has only the role of fertilization. Then, when the environmental signals become less important, copulation can have two functions, namely causing ovulation and fertilization at the same time. And then later, there is an evolution of a decoupling between copulation and ovulation. Ovulation is then internally determined by the physiology of the female. And that's what I call spontaneous ovulation. Spontaneous in the sense of not determined by extra-organismal stimuli. Now, spontaneous ovulation, at least according to this analysis, seems to have evolved from induced ovulation. So here, spontaneous ovulation, all the black lineages in a simplified phylogeny of eutherians, including marsupials here. And here with the red arrows, I indicate the times in the phylogeny when menstruation evolved. And you can see in either case, the menstruation evolved deeply embedded in a clade, in a group of animals that have already spontaneous ovulation. I have no explanation why that is important for menstruation, but that seems to be one of the patterns that is relatively well supported. Finally, within the species with spontaneous ovulation are species that have spontaneous decidualization. I will explain this in a second. And it turns out, and is actually a point where the story is getting a little bit messy, that a lot of species that menstruate also have spontaneous decidualization. But it turns out that is not a perfect match, because there are also species that are have no clear signs of spontaneous decidualization, but nevertheless have been described to be menstruating. So decidualization, of course, is this differentiation of the endometrium and the embedding of the fetus or the placenta, and this decidual cell and the decidua in general, including the leukocytes that are part of it, are an essential player in embryo implantation. Now, there are two forms of decidualization. In most animals, decidualization is embryo-dependent, meaning that the blastocyst or the embryo, as it arrives in the uterus, is sending out signals that then lead to the differentiation of the endometrium. Example is, for instance, the mouse. Or the spontaneous decidualization, where the decidual cells differentiate, the whole tissue becomes the decidua, even before or whether or not an embryo arrives as part of the sexual cycle. So decidualization is part of the sexual cycle and independent of the presence of an embryo. Some people caution and say that what we see here is maybe a precursor of decidualization, but clearly in some animals the endometrium is preparing for the arriving embryo before the embryo is even there. And that is what we, of course, know from humans and many other menstruating species. So I told you earlier that falling progesterone levels lead to tissue destruction and therefore to menstruation. But there is an interesting point here to make, and that is the following. In mice, lab mice, they are having a very short ovarian cycle. And if there is no copulation during estrus, there is essentially an immediately return to the follicular phase, and there is no proper luteal phase with the production of progesterone. So I told you that progesterone withdrawal leads to inflammation menstruation, but if we have a mouse that has pseudopregnancy, for instance, when you mate the mouse with a vasectomized male, copulation alone is then sufficient to maintain the corpus luteum, and then the mouse after pseudopregnancy, progesterone levels decrease, but it does not alone leads to bleeding and menstruation. But there were a number of attempts to synthesize sort of a mouse model of menstruation, going back all the way to the work of Finn and Pope in 1983. And this is an experimental paradigm that has been refined and reworked many times. One of the more recent papers that I'm aware of is this paper by Isabel Gasho in 2012. Now the experiment goes like this. You take a female and mate it with a vasectomized as male, so there will be no fertilization, but there is mechanical stimulation of the cervix and the vagina and the clitoris, which is sufficient for maintaining the corpus luteum, so that the progesterone level will increase. And 4 days after successful copulation, the female is injected in the uterus with a droplet of oil, sesame oil or any other irritant, which is known to be able to cause a decidual reaction. It's called a deciduoma. It's a question to what degree this actually reproduces decidualization or only partially. But anyway, in this experiment, it is used as a way to decidualize the endometrium, and then the animals are monitored, and then here you see the progesterone level is increasing from proestrus, estrus into 4 or 5 days, and then decreasing towards 9 and 10 days. And as the progesterone levels are falling, there are more and more incidences of heavy bleeding with visible blood flow, blood outside the vagina, and so on. So now this experiment shows that only after decidualization, falling progesterone levels leads to tissue destruction and menstruation. In other words, the combination of luteal phase with then decreasing progesterone levels and preceding decidualization is sufficient to cause a situation that seems to reproduce what we know as menstruation. Now, this is an idea that was then further developed by Colin Finn, that menstruation as this bleeding event is an epiphenomenal and non-adaptive consequence of uterine evolution. And what he means is evolution of spontaneous decidualization. So why would spontaneous decidualization be a good idea biologically? So Finn has argued that the differentiated endometrium has the function of both enabling and also regulating the degree to which the placenta can invade into the maternal tissue. Normally, the myometrium, the muscle part, and the placenta are separated by an endometrial layer of decidua. But if the endometrium is not able to contain the invasion of the placenta, it can lead to a quite serious situation, either an attachment to the myometrium, or even an invasion of the myometrium, or even a perforation of the uterine wall into the peritoneum. Now, these conditions, placenta accreta, increta, and percreta, without medical intervention, are usually fatal to the mother after birth, because the bleeding after the removal of the placenta usually is stopped by the contraction of the spiral arteries in the decidua. But if the decidua are not there, the myometrium doesn't have spiral arteries, and therefore has a hard time stopping the hemorrhage. So it's obviously clear that preventing or decreasing the frequency of placenta accreta, or even worse cases, is a good idea. And Finn's idea was that you evolve spontaneous decidualization to avoid an aggressive fetus to be able to reach the myometrium during the implantation and and development of the placenta. Now, we actually also produced some in collaboration with my friends in the biomedical engineering department, and quantitative assay for invasiveness that consists of these nano-structured surfaces that are just the right size, that they mimic collagen fibers. And because we all know that cells love to travel along collagen fibers, so with this contraption here, you can make cells move in one direction rather than in all directions. Now, if you co-culture an invasive cell population next to a stromal cell population, let's say trophoblast cells, extracellular trophoblast cells, and let's say endometrial stromal cells, you can then measure how much and how quickly the invading cells are able to invade the domain of the stromal cells. Now, and if we did these experiments with undifferentiated human endometrial stromal cells, you see here that there is an increase in the distance in which they are able to travel within 3 days. But if you, instead of using human endometrial stromal fibroblasts, decidual cells, in vitro decidualized cells, the invasion is much more limited. Here you see it quantified. In the white is the control with non-decidualized stromal cells, and you can see over 3 days how the spread of the trophoblast cells is increasing. Is also increasing with decidualized cells, but to a much lesser degree. But all of that sort of makes sense, that decidualization is a good idea. Do it early to make sure that you're ready for the fetus to come and invade maternal tissue. So this idea by Colin Finn, that spontaneous decidualization is a protection against invasion by the trophoblast, because decidual enable and limit trophoblast invasion. Spontaneous decidualization makes sure that the uterus is prepared before the blastocyst arrives in the uterus, and that should decrease the risk of damaging invasion and thus protect the life of the mother. However, the problem is that if you look very carefully in the literature, you discover many species that are described to be menstruating but are not spontaneously decidualizing, at least based on the criteria that people have used over the years using histology. So here, red are rodents, black is the Afrotherians, the green species names are bats, dark blue is Old World monkeys, and these are New World monkeys. And you see that there's quite a number of these menstruating species that have no obvious signs of spontaneous decidualization, meaning no decidualization before the arrival of the embryo. There are 2 of the bats, and in particular 2 Old World monkeys, which is a particular head scratcher because they are relatively closely related to apes and humans, and should be our closest cousins doing the same as we do. So here is what is going on with the rhesus monkey, as what's called a superficial implantation or attachment, if you want. So here's the blastocyst, here's the endometrium epithelium, and here's the endometrial stroma. And the authors describe that there is no endometrial decidualization of the stroma. Instead, what is happening after the attachment is that the epithelium develops this epithelial plaque here, that is this dark region here. So the epithelium is proliferating, and the embryo or the blastocyst is not even interacting or nowhere close to the endometrial stromal fibroblast cells, and they remain quite superficial, even all the way to, let's say, 14 days post-copulation. And that's possible because there's a superficial system of blood vessels that breaks through the epithelial plaque here and perfuses the placenta. So a long time into this gestation of the rhesus monkey, there is no direct interaction with the stroma, the endometrial stroma, and correspondingly also no spontaneous desitualization or even immediate desitualization. It only develops later when the placenta is embedding deeper into the endometrium. So the relationship between spontaneous desitualization and menstruation is somewhat complicated. Menstruation occurs often in species with spontaneous desitualization. And actually the question is whether there are any species with spontaneous desitualization that do not menstruate. I don't know of examples. If anyone knows, I would be happy to learn about it. So in other words, it would mean that spontaneous desitualization is sufficient for menstruation, as for instance shown by this mouse experiment, but it's not necessary. And that of course leads an open question of what is then necessary, or what are alternative ways that you get menstruation, like in the old world monkeys. So as I told you, there are these species that menstruate but have no clear evidence of spontaneous desitualization. And of course, there are various possibilities. One is that it might be hard to detect desitualization in different species, for instance, because marker genes for the differentiation of the decidual cells can be species specific. For instance, prolactin, one of the classical human markers for human decidual cells, is regulated by different cis-regulatory elements in different species, and therefore most likely independently evolved in rodents, in primates, particular apes, and in other species. So it's clear that the desitualization differentiation markers can differ. Maybe also the histological signs could not be as clear-cut. That could be a problem. Why maybe desitualization is not recognized, even though it might be there in a physiological sense. Or there are other transformations of the endometrium which prepare the endometrium for the embryo, that are not recognized as desitualization in the technical sense, but nevertheless have the consequence of leading to menstruation after progesterone is withdrawn. Okay, now let's talk about what theories exist about the biological role of menstruation. One we already talked about, this is this idea that this menstruation is a side effect of spontaneous desitualization and so on, and as I have already explained. There are three other ones that I want to mention. One that is gained a lot of notoriety and attention is the idea of Margie Prophet in a paper in 1993, where she argued that menstruation is a defense against pathogens transported by sperm. The career of Margie Prophet and her struggle to understand menstruation made it all the way to the theater stage. There's a play about Margie Prophet's career and life. Now, what the question that I have with this theory is that I don't see that the 96% of Eutherian species out there wouldn't be as vulnerable to the introduction of pathogens during copulation as the 150 species that are actually menstruating. So I find this a very interesting and creative idea, but I don't think it is very plausible from a comparative point of view. The next theory, which was actually published in response to the pathogen defense theory, is that by Beverly Strassman in a paper 3 years later, 96, proposed that endometrial cycling and menstruation is a way to save energy, because because the maintenance of a fully differentiated endometrium would cost too much energy. And again, my question is, why would that only apply to the small minority of species that have menstruation and not to others as well? And in particular, because there are other ways of reducing the amount of endometrial tissue outside the reproductive period, and that is what is happening with many, many species. So I don't think, based on comparative evidence, it's not very plausible. The next one is the theory of embryo selection, going back to the work of Jan Brosens, University of Warwick. It was a relatively new idea. And this is based actually on some clinical observations that Dr. Brosens has performed, as, for instance, summarized in this paper from 2010. So what he observed is he had patients with recurrent pregnancy loss, and was working with them. And at some point, he was noticing that the interval between the last pregnancy loss and the next clinical pregnancy is much shorter in patients with recurrent pregnancy loss than it is with the normal population. And this is also correlated with a higher expression of receptivity markers, and most relevant here, a lower expression of decidualization markers. And so the theory goes that early embryos differ greatly in terms of their viability. Maternal investment in low-quality embryo carries a higher cost for the mother. So spontaneously, decidualized cells require a robust signal from the embryo to accommodate the embryo and prevent luteolysis and abortion. So what is happening is that properly differentiated decidual cells are only supporting embryos that are robust enough, strong enough to prevent luteolysis, and therefore they are a selection mechanism for embryos and selecting for high quality blastocysts. So therefore his theory is that spontaneous decidualization evolved to provide a stronger selective environment for the embryo and to protect the mother against futile investment. Of course, this is a theory that fits well with the human and great ape evidence. But again, we have the same problem of that spontaneous decidualization is not strictly associated with menstruation. And maybe there's generally an association between higher offspring quality and the need to select for higher offspring quality in one way or the other that is correlated with menstruation, but not necessarily spontaneous decidualization, as we have seen. And now this idea has been supported in part also by the observation that the menstruating species like higher primates, bats, elephant shrew, all have very small litter size, either one, usually one or two. And in this whole picture, also the spiny mouse fits quite well, because it's usually around 2 offspring per pregnancy in the case of the spiny mouse. So let's have a closer look at the spiny mice and compare them with other laboratory rodents. And here you see a portrait of the first litter of spiny mice born at Yale in our colony. And if we compare the spiny mouse Acomys with an average litter size of about 1.7 on average with that of, let's say, the Mongolian gerbil, house mouse, and the rat, it looks like this is a quite substantial decrease in smaller, at least substantially smaller litter size. Now, in order to see whether that's a coincidence or a systematic pattern, I asked an undergraduate student 2 years ago to collect data on litter size of rodents, and she found data for about 1,004 species, which is about half of the total rodent diversity, and map litter size onto the phylogeny of these 1,000 species. And you see the species tree here. And Gladys is the student who did all the hard work. Now, if we take the data that she has gathered and just look at the distribution of litter size reported in the literature for these 1,004 species, we get about this distribution here. And the spiny mouse is somewhere here. The average is about 3.5, or the median is 3.2. The mouse sits somewhere between 5 and 6, and the rat with 7 about here. But we have to recognize that among the 1,000 species, there are 173 species and so meaning 17% that have an average litter size between 0 and, of course, 0 is not larger than 0 and less than 2, which is 70% of this taxon sample. So, of course, it's possible there are many more rodents that have that are menstruating, but it's not known that any of those are menstruating. And it took the research community actually quite a number of decades to figure out that the spiny mouse is actually menstruating. So there's a possibility that there are more out here, but I would be surprised if it is actually 17% of all rodents that we haven't been able to recognize that they are menstruating. We can also compare it not to the species level, but also reconstruct phylogenetically what would be the litter size of the rodent ancestor. And according to this reconstruction, it's about 3. So that means that the contrast between lab rodents and the spiny mouse is a little bit not typical, is a little bit larger than what is actually the situation for Acomys. So Acomys is about 40% less than the ancestor, and maybe half of the average litter size that we know of other rodents. So if we look at the spiny mice and compare them with these animals here, they do have a smaller litter size, maybe 1/2 of compared to them, and they have longer gestation times, 39 days compared to between 20 and 30 or 19 or 20, and also give birth to quite developed offspring with open eyes and furry compared to the gerbils or the mice that we of course all know more or less. Now, the comparison between the gerbils and the spiny mouse is actually quite interesting because, as I will show you in a second, the gerbils are very closely related to the spiny mouse. So we can infer that, in fact, the gestational length increased relative to its outgroup, and the average litter sizes was decreasing. So if we have here this phylogeny of 1,000 rodent species, and we zoom in into the part where we have the spiny mice, then we see this here. So here we have the spiny mice, and if we trace back the phylogeny from the spiny mice back in time, we are coming to a node here where then the closest clade, the closest group, then leads us to the gerbils. So gerbils are the sister group to spiny mice, and therefore are an excellent comparison to investigate how menstruation in Acomys in the spiny mice may have evolved, because they are very closely related. So we started a colony of Mongolian gerbils in our lab and started to look into the endometrial biology in gerbil ovarian cycle, and looked at all of the different stages. And to our surprise, what we found in diestrus, where this is after ovulation and is roughly comparable to the luteal phase in the menstruating species, we found cases where there's quite extensive desquamatation of the luteal epithelium, and in the lumen we find debris and erythrocytes. Now that is not a huge hemorrhage, but it is still interesting. Here, mucin as an epithelial marker shows that this is actually lacking an epithelium here. Cytokeratin can also be used for that. Interestingly, we also found a lot of cases of this metaplagic morphology, and that is not just from an oblique section. We looked into this very carefully. There seems to be a lot of sort of metaplastic lesions in the luminal epithelium of this. So here you have another animal with quite extensive of these proliferations of the epithelium. Here's an example in proestrus, probably a case of re-epithelialization here with cytokeratin. And of course, we also look into single cell analysis, but this is an ongoing work. I'm just giving you a little bit of a taste of where this is going. Here, and specifically looked into the mesenchymal clusters, and one of which is clearly contractile cells, and they are only found around the blood vessels and in the myometrium, maybe here in the transition zone somewhat, but not in the myometrium. This is smooth muscle actin immunohistochemistry. But so this is the contractile part. This is probably the cluster of endometrial stromal fibroblasts with one subpopulation that is particularly interesting because it has a lot of cells expressing Wind4 and fibroblast activating protein. So that seems to be a subpopulation of endometrial stromal cells that have undergone some kind of transformation, maybe a fibroblast activation or some precursor to pre-decidralization as indicated maybe by Wind4. Okay, so one last point here, and that is not only has the spiny mouse evolved a longer gestation in a smaller average litter size, it also has re-evolved luteal phase in its ovarian cycle. Now, as I already mentioned before, in most rodents, there is no real luteal phase, because if there is no fertilization, the next follicular phase starts immediately, and there is in diestrus no progesterone expression as seen, for instance, here in gerbil. There is this reported high expression in estrus, but I'm not sure I believe it given the large error bars here. But in the spiny mouse, in the data from Bella Fiore, the diestrus has a very marked increase in progesterone production. And so we have to take into account that what's happening in the evolution of the spiny mice was a re-evolution of the ovarian of the luteal phase in a rodent. And I thank my colleague and friend, Professor Pavlichev, who pointed out this fact to me, which was important for my thinking to click into register. So that all sort of can lead to a scenario, at least for the spiny mouse evolution. We recognize there's always a phase of endometrial remodeling in diestrus, to some degree in mice even, but even a little bit more dramatic in the gerbil, the closest relative to spiny mice, is desquamatation and metaplastic lesions in the luminum epithelium. Putative activated decidual fibroblasts or the endometrial fibroblasts, and most rodents suppress the luteal phase, and in spiny mice it has re-evolved. So maybe what we can argue is that there leads to a model where the luteal phase accelerates and deepens the endometrial remodeling, which is already present even in gerbils, and then eventually leads to the tissue breakdown and overt bleeding as recognized as menstruation. It's also, from an evolutionary point of view, interesting to note here that what in spiny mouse is not just a re-evolution of the luteal phase, but actually the loss of the suppression of the luteal phase. And this can, of course, be due to some non-adaptive causes, either by simple degeneration and small population size in these desert animals, or a side effect of the evolution of longer gestation, or whatever. So it doesn't need to be an adaptive evolutionary event. So with that, I want to summarize quickly. So the evolutionary origin of menstruation is still difficult to explain, primarily because there's no very neat, consistent set of specific biological traits that is associated with menstruation. Already talked about the lack of strict correlation of spontaneous dehydrogenation. Also, a lot of people have argued that spiral arteries are sort of the anatomical cause of menstruating, but there are New World monkeys where explicitly spiral arteries were not found. Litter size can be an issue, but it's actually small litter size is much too frequent to be a very strong predictor of menstruation. But there is an association with lower litter size and long gestation, and thereby an association with increased investment in offspring quality, which would support the notion or go into the direction of the model by Jan Brosens that it has something to do with embryo selection, either by spontaneous dehydrogenation or other mechanisms that would be happening in other species that don't have spontaneous dehydrogenation. And it's also to note that this is only happening in animals with hemochorial placenta, so meaning that like cattle has also long gestation and small litter size, but because they have epithelial coral placentation, they probably are not developing this phenomenon. So with that, I thank you for your attention, and just want to mention that the next talk will be October 19th by Antonis Rokas from Vanderbilt University, and he will talk to us about the evolutionary regulation of gestation length. Thank you very much for your attention.